Have you ever wondered how our cells duplicate their DNA? It's a question that has been at the forefront of scientific inquiry for decades, a mystery that has been slowly unraveled through the meticulous work of countless researchers. This is the world of DNA replication, a process as intricate as it is essential. At the heart of every cell in our bodies, DNA replication is a marvel of biological engineering. It's the process that enables life to persist generation after generation, allowing our cells to divide and reproduce with stunning precision. It's a marvel of nature's microscopic craftsmanship, a dance of molecules choreographed with breathtaking complexity. From the opening of the double-stranded helical structure of DNA and separation of the strands through the priming of the template strands to the assembly of the newly formed DNA segments, DNA replication is a journey into the very essence of life. Let's dive into the fascinating world of DNA replication. DNA replication, in essence, is a three-step dance. It's a meticulously choreographed performance that occurs within every living cell, ensuring the continuity of life. First, we have the opening act, where the double-stranded helical structure of DNA parts ways. This separation occurs at a specific site known as the origin. Picture two entwined dancers taking a step back, creating space for the performance to unfold. This opening of the DNA structure is facilitated by several enzymes and proteins, each playing their part in the grand ballet of life. Next up, we have the priming of the template strands. Now that the DNA strands have been separated, they need to be prepared or primed for duplication. This priming is a critical step as it sets the stage for the creation of new DNA segments. It's akin to the dancers getting into their starting positions, ready to perform their routine. Finally, we reach the crescendo of our performance, the assembly of the newly formed DNA segments. Here, an enzyme called DNA polymerase takes center stage. Its role? To organize the assembly of the new DNA strands. Like a master choreographer, the DNA polymerase ensures each new DNA segment is in its rightful place. Of course, it's worth noting that these steps aren't rigid. The dance of DNA replication can have slight variations depending on the organism and cell type. However, the core steps, opening, priming, and assembly, remain consistent across the board. Enzymes and proteins are the unsung heroes of this process. They catalyze several important stages of DNA replication, ensuring that the dance goes off without a hitch. From facilitating the initial opening of the DNA structure to organizing the assembly of new DNA strands, enzymes and proteins play a critical role in the grand scheme of DNA replication. So, the stage is set, the players are ready, and the dance of DNA replication begins. Stay tuned as we delve deeper into the role of enzymes and take a closer look at DNA replication in E. coli in the next scenes of this captivating biological ballet. Enzymes are the unsung heroes of DNA replication. This statement isn't an exaggeration, but a testament to the vital role enzymes play in the intricate process of DNA replication. Enzymes are essentially the conductors of the DNA replication orchestra. They regulate the tempo, direct the performers, and ensure that every note is perfectly executed. Without them, the beautiful symphony of DNA replication would descend into chaotic cacophony. Let's delve deeper into how these enzymes work their magic. The opening act begins with the enzyme helicase. This molecular motor wrenches apart the two strands of the DNA double helix at the replication origin, creating a replication fork. This is akin to unzipping a tightly fastened zipper, only infinitely more complex. Next in line are the single-strand DNA binding proteins, or SSBs. These enzymes stabilize the separated strands and prevent them from recoiling back into their double helical structure. It's a bit like holding the ends of the unzipped zipper apart. The stage is now set for the primase enzyme. Primase lays down a short RNA primer on the DNA template strand, a necessary step that allows DNA polymerase, the star of the show, to take the stage. DNA polymerase cannot initiate the synthesis of a new DNA strand from scratch. It needs a primer to which it can add DNA nucleotides, and so enters DNA polymerase. This enzyme takes center stage, adding nucleotides to the RNA primer. It moves along the template strand, synthesizing a complementary strand of DNA, but the performance isn't over yet. Another enzyme, DNA ligase, steps in to seal any remaining gaps between the newly synthesized DNA fragments, ensuring a continuous DNA strand. Think of it as the final bow that brings the whole performance together. Without these enzymes, DNA replication would be impossible. They catalyze critical stages in the process, helping to organize the assembly of new DNA strands. Each enzyme has a unique role to play, and together they form a flawless ensemble, executing a complex process with precision and finesse. These enzymes ensure that the dance of DNA replication is perfectly choreographed. From the unwinding of the DNA helix to the formation of new DNA strands, enzymes are indeed the unsung heroes of DNA replication. Let's take a closer look at DNA replication in E. coli. DNA replication in E. coli is a fascinating process, with a mechanism that's strikingly similar to what we see in eukaryotic cells, those cells with a nucleus that make up organisms like you and me. In E. coli, a bacterium that resides in our gut, the replication process gets kick-started at a precise location known as the ORIC locus, or ORIC for short. This is where the DNA protein takes center stage. Binding to the ORIC, DNA has the crucial task of initiating the replication process. As it binds, it also hydrolyzes ATP, a molecule that provides the energy for many cellular processes. This sets the stage for the replication machinery to move in and start the delicate task of duplicating the DNA. Even in the smallest of organisms, the process of DNA replication is a marvel of nature. DNA replication is more than just a biological process. It's a symphony of precise steps, harmoniously conducted by various enzymes and proteins, each playing their part to perfection. This fascinating dance of molecular interactions begins with the opening of the DNA's double-stranded helical structure. It continues with the priming of the template strands and concludes with the assembly of new DNA segments orchestrated by the maestro DNA polymerase. The process is universal yet uniquely tailored to the organism and cell type in question. Take E. coli for instance, where DNA replication is initiated at the ORIC locus, demonstrating the specificity of this process. DNA replication is a testament to the marvel of cellular function. It underscores the importance of every single cell in our bodies, each undertaking this intricate process moment by moment. Understanding DNA replication helps us appreciate the intricate and beautiful dance of life that happens inside each of our cells every moment.